Okay, today we're going to talk about Necrobiosis lipoidica. There's actually been several recent papers on this. I'm just going to talk about my work because I didn't incorporate those recent two publications. I do have a conflict of interest. I have a small biotech company called PhysioSeek. It doesn't have anything to do with this presentation, though. Necrobiosis lipoidica is one of the classic dermatologic diseases. Yet we don't know much about it. It was first described in 1929 as dermatitis atrophicans diabetica, and then in 1932 it got its name that we still use today, Necrobiosis lipoidica diabeticorum. Eventually, non-diabetic patients were found, and then they eventually just dropped the diabetes part of it. And today we call it necrobiosis lipoidica. But in clinic, I sometimes still say NLD because it just sounds better. Okay, so what are some facts about NL? Well, it's a chronic granulomatous disease. There's a number of NL cases that have diabetes, but it's not the majority. Only about like 1% of diabetic patients get it. It's more common in type 1 diabetes than type 2. It's not really proven to be connected to glycemic control. So if you get your patients under better glycemic control, it doesn't necessarily mean that they're going to get any better. It's associated with other diseases like sarcoidosis, but maybe that's just because they're both granulomatous and sometimes it's hard to distinguish. Average age is 25 if you have diabetes and 46 if you don't have diabetes, and female is greater than male incidence. Um, it's it's one of our classic diseases. So if you're a dermatologist, you could diagnose this without biopsy. But for those of you who don't know, patients get these indolent red, yellow, brown atrophic plaques, predominantly over over their pretibial lower legs. These lesions can also have telangiectasias, and they could also have an inflammatory rim, which shows that they're actually growing in size. They could have cavernization, and they could ulcerate. Like if you do a biopsy on it, it's possible that you're going to cause it to ulcerate, and then ulcerative NLD is a little bit difficult to treat as well. Some people think that they could occur in atypical places like upper extremities, face, and scalp. There's a related disease that looks very similar on pathology called necrobiosis xanthogranuloma, which is much more common on the upper extremities and face. There's no randomized controlled trials for NL. So although this is one of our classic dermatologic diseases, we don't really know much about therapy. There's been lots of treatments tried in like case reports. New kids on the block are like the JAK inhibitors. Those seem to be working pretty well. Ustekinumab, anti-TNFs are kind of like the gold standard for therapy. People also use topical steroids, methotrexate, and like since it's been around since beginning of time, there's all these old therapies that are being used for it as well. There's a recent report of topical ruxolitinib, which is Jakafi if you take it orally. That seemed to work okay. They had a 58% improvement in their disease score. But this is prospective, but not randomized, and these disease scores aren't like highly validated. So some real life cases. These are some cases from me. This is 4 months of secukinumab. Didn't really do anything. So from this one patient, I could say that IL-17 doesn't work inhibition. Ustekinumab, I actually use ustekinumab in some patients. This patient didn't respond initially, but then after like years, they became responsive, and it seems to not be in remission, but seems to be well controlled. I typically use anti-TNFs. So this is a patient who is treated for ustekinumab for a few months, and then we switched to anti-TNF, and you can see a marked improvement in her lesions. You can see some older lesions turning dark, but she still has active disease after 3 months, and that's after being on ustekinumab for 3 months. So I think TNFs are still my go-to therapy in this disease. This is 4 years of adalimumab, and this patient has chronic stasis dermatitis and obviously other things going on, but you could see that from initial presentation that was highly ulcerative, there is some improvement. The JAK inhibitors are becoming more and more common in clinic. These block signaling from interferon gamma, IL-13, IL-6, IL-12, and IL-23 receptors. They have been used in NL as well, not with like great success, but it seems to be very promising. So here's a patient who's been on 6 months of tofacitinib. This is a patient who received ruxolitinib for cancer, and it looks like their NL got a little bit better. This is a patient on baricitinib. These are not my patients. These are published case reports. Here's a nice study that showed some effectiveness of tofacitinib twice daily. Patient still has some disease. This is another patient treated with tofacitinib, but they got at 9 months, they were also getting interlesional corticosteroids. So it might not be a bad idea to do some type of combination therapy for these patients. So in summary, although many agents have shown efficacy in NL, there are no highly effective treatments for NL. Remission on therapy remains difficult to achieve, and there's no randomized control trials in NL. So what do we know about NL pathophysiology? Recently with these papers, we know a little bit, but prior to these papers that came out late last year and this year, we didn't know much. We knew that the epidermis looked pretty normal or slightly atrophic. It had this the biopsies have this rectangular appearance, which is common in sclerosing diseases. It has this hallmark layer caking pattern, which you kind of could see here, where you have fibrotic areas and necrobiotic areas surrounded by areas of inflammation. Blood vessels could be thickened, and there are plasma cells, especially in the deep dermis. So what is necrobiosis? And necrobiosis doesn't have a definition. It's more of something, appearance on a slide. There's no like definition of necrobiosis. And if you go into different textbooks, they use wildly different things to describe it. Like this textbook is calling it dead collagen. I don't know if you can see that here. And then if you look in these common textbooks, they're all different definitions. But clinically, we know that, or histologically, we know that it's pale staining smudged collagen that looks possibly necrotic. Some people say it's red in collagen. Some people say that there's loss of collagen bundle integrity and nuclear debris. So, and then I'm not going to talk about NXG, but my recent publication also goes into NXG. NXG is very similar to necrobiosis. You just have a few more prominent macrophages and giant cells, and you have cholesterol clefts, and the pattern is not really layer caked. It's more of this X pattern, but it's very similar. You can see this very large giant cell here. Very similar in terms of pathophysiology. Okay, so how are we going to study this so I can explain to you what necrobiosis is? There's two ways to do RNA sequencing these days. One is bulk RNA sequencing, and that's how we made all of our nice discoveries, initial discoveries in psoriasis and atopic dermatitis. And then there's the new kid on the block, which is single cell sequencing. And you can think of bulk sequencing like a smoothie. You just put all the cells together, you blend it up, and you look at the gene expression pattern. And you could also do some complex analysis to kind of deconvolute what types of cells were there after you do this analysis. Or you could just keep the cells separate and do single cell sequencing. Each technique actually has advantages. So one technique doesn't replace the other. Single cell sequencing is good because you keep the identity of the cells intact. Bulk sequencing is good because it detects the low abundant transcripts that might not be easily detectable in single cell sequencing. So we do both techniques, and then later at the end of the talk, I'm gonna explain spatial sequencing, which is the real new kid on the block, and that is gonna totally change our understanding of all dermatologic diseases. Okay, so this is just the same thing. In single cell sequencing, you keep all the cells separate, and then you could do some expression profiles of the individual cells. And in bulk sequencing, you just mix everything together. It's nice to do single cell sequencing because the skin has a ton of different cells at different layers, and Mixing all of them together and trying to figure out what's going on is a lot harder than keeping them separate. So we usually start with bulk RNA sequencing, and that's what we did with NL. And this identified that there was a strong upregulation of some chemokines, namely CCL5 and CXCL9. There's a very high upregulation of IgM. And then we started getting very high expressions of some collagens that are usually expressed at low levels in the skin, like collagen 4A, 4. We were then able to do some deconvolution analysis from this. That means like take those bulk signals and try to figure out which cell types are in the skin. And we found increased macrophages, B cells, cytotoxic T cells. And then we did some immunohistochemistry to show that these chemokines, and we did a lot of immunohistochemistry in this paper, but basically from immunohistochemistry, you could kind of identify different cell types by H&E, by their appearance, and then you could see what they're expressing at the protein level by using antibodies that are specific to those proteins. And we're able to see CCL5 in lymphoid cells, fibroblasts, and macrophages, and we're able to see CXCL9 in fibroblasts and macrophages. We also did deconvolution analysis. Deconvolution analysis also identified elevated neutrophils and T cells, but not Th17 cells, which is probably why that one patient didn't respond to secukinumab. But then we switched over to single cell sequencing to get a better idea of what's going on. This is our single cell sequencer. There's many different platforms. We use the 10x Genomics platform because it provides a much deeper capture of the transcriptome. So we're not interested in sequencing more cells if we're not capturing a large portion of the transcriptome. So when we first started doing single cell sequencing, we were only sequencing about 7% of the transcripts. And they just released new chemistry a few months ago, and now we could sequence up to 50% of the transcripts in the cells. So this is rapidly evolving technology. That means that the papers we published, 3 years ago, are not nearly using advanced technology that we use today. So single cell sequencing, one disadvantage is that you don't sequence a lot of cells. You sequence, with this technique, you sequence about 20,000 cells. With other techniques, you can sequence more cells, but the number of transcripts per cell is less. This requires you getting a single cell preparation of highly purified cells, and then you put them through this flow cell, and they get incorporated into a droplet, and then all of the things that you need to do RNA-seq with gets inserted into that droplet. And these droplets are very stable, so you have basically like 20,000 different reactions going on. Everything is barcoded, so you could go back and figure out that these transcripts came from a single cell and that they weren't from multiple cells. We did this in necrobiosis, and this is a very complex analysis, but you could flatten it through dimensionality reduction, and there's several strategies to do that, but you could look at this really, really high parameter data as a two-dimensional image. Like here, this is a two-dimensional image. This is called UMAP. This method is called the UMAP method. And from there, the cells that are similar to each other cluster together. So these cells that are all in a cluster, they're usually the same type of cell. And then you could use classic cell markers to identify which cells they are. So you can identify the fibroblasts by their expression of classic fibroblast markers, keratinocytes, T cell, B cells, myeloid cells. And from just this analysis, you could see that lesional, so we biopsied non-lesional skin and lesional skin. And of course, this is, kind of difficult for the patients because they could get ulcers where you biopsy. And when we did that, you could see that lesional skin has this huge expansion of T cells and a huge expansion of B cells. And then fibroblasts look like they're compressed here, but that is the fibroblasts compared to normal. That's not because there's fewer fibroblasts. This is just a percentage-wise. So now that you have an infiltrate of all these cells there, percentage-wise, fibroblasts make up less of the lesion, but they're still very, very important, as we will discuss. So first we looked at these fibroblasts, and if you remember from the bulk sequencing, we know that we had CCL5 and CXCL9 in fibroblasts from the immunohistochemistry. And now we could really identify the different fibroblast populations. This is a heat map of all the clusters of, so when we look at fibroblasts from the original UMAP, and then we just cluster these cells together. So we're just looking at the fibroblast population. And then you could, since we mixed this with normal non-lesional cells, we could see that there's a new population in NL. This kind of horn here is only present in NL, and those are cells expressing fibroblasts expressing CCL5 and CXCL9. And then we could characterize those cells more because we know their entire transcriptome now, and we know that these are like CD74 high. And also interesting, they're expressing high levels of collagens that aren't usually expressed in the skin, or at least not at high level. So they're expressing a lot of collagen 11A1, a lot of collagen 8A1. So in addition to there being more collagen present in these necrobiotic lesions, it's actually not normal collagen. You're getting other collagen molecules being deposited there. And there's also a lot of FN1 fibronectin being deposited as well. Interestingly, the fibroblasts also are a good source. These CCL5 fibroblasts are also a good source of IL-32. When we look, since CCL5 and CXCL9 were both secreted by these fibroblasts, I thought naively that they would have the same stimulus to cause the fibroblasts to secrete them. But as you could see here, these are cultured fibroblasts, and then we in vitro culture them with different stimuli, and you could see that CCL5 is secreted in response to different stimuli than CXCL9. But CCL5 is really responsive to TNF, which might be one reason why blocking TNF is helpful. And then these fibroblasts are CD34, they're IL-32 positive, but these collagens don't seem to be responsive to these stimuli. So there must be something else driving the collagen 4A4 production. And there's also different matrix metalloproteinases that are being secreted. These are enzymes that degrade collagen, and they don't seem to be responsive to any of these stimuli. Okay, so then we looked at macrophages, and it turned out that there's this SSP1 high. So these macrophages were new in necrobiosis lesions that we didn't see in healthy skin. And these were expressing this FC gamma R3A receptor, and they're expressing very high SPP1. We could take the myeloid cells and do a heat map of all the different clusters, and they clearly express different genes. We could use some marker genes to kind of better identify them. Marco is one of these genes of macrophage differentiation, and that it's highly expressed in these SPP1 high macrophages. SPP1 is the gene that encodes osteopontin, and we'll talk about that osteopontin later. But this is the gene that has multiple biological functions, including in the immune system and outside of the immune system. So far we have CCL5 fibroblasts, SPP1 macrophages, and then when we looked at the T cells, there was another unique population of T cells that were dominant in necrobiosis, not present or very few in normal skin, like 3% of T cells in normal skin are NKG7 high T cells. There are tons of T cells in necrobiosis, so it's not just NKG7 high T cells. But since NKG7 high T cells don't usually represent a lot of your healthy skin, it's pretty evident that they're doing something here in necrobiosis. We'll talk about them more in another slide, but these T cells are also secreting CCL5, and they're CD8 T cells, and they're secreting lots of CD8 effector, or at least they're expressing lots of CD8 effector molecules like granzyme, perforin, etc. And when we do these studies, so down here, this is scleroderma. So we used other diseases. So I'm not presenting that data due to time, but we compared these results to those observed in other diseases. And the two model diseases that we used to compare necrobiosis to were scleroderma, systemic sclerosis, and sarcoidosis. Sarcoidosis being an example of a granulomatous disease, and systemic sclerosis being an example of a collagen deposition disease. And one weird thing, which actually has been possibly reported in another dermatologic disease, is that necrobiosis had a lot of IgM expressing plasma cells. So usually, if you remember your immunology class, you have your naive B cells, those express like IgD on their surface, and those undergo class switching to IgM. And then with additional antigen stimulation, those undergo class switching to like IgG, IgA, and IgE. You're not supposed to have IgM plasma cells, but necrobiosis is chock full of these IgM expressing plasma cells. There's also IgG expressing plasma cells, so they're not the only plasma cells there. But here in this whole corner here, these are all IgM expressing plasma cells. These are the naive B cells, and these are the IgG expressing plasma cells. Okay, so this is another technique that's very similar to single cell sequencing. And we did this on, and the advantage of this technique is you could do it on archive specimens. And the single cell technique is fairly robust from these archive specimens. It uses a different technology, but it's still, we still use our same instrument to do it. And these experiments were done by University of Michigan. And we were able to find through archive tissue that NL and had tons of IgM plasma cells, but these weren't, and they're also in NXG as well, but they're not present in other skin diseases like systemic sclerosis. The other thing that we're very good at is we're very good at integrating data. So there's also single cell RNA-seq, but there's single cell T cell receptor and B cell receptor sequencing. We could capture nearly all the T cells, T cell receptor sequences and B cell receptor sequences. And the reason why that's interesting is because as the cell divides, single cell sequencing will tell us that what's in that individual cell. But as the cell divides, you're going to get more copies of it. So you don't know if it's a clonal expansion or if it's just multiple copies of just single unexpanded T cells. And we're able to do single cell TCR and BCR sequencing, and on the same cell that we're doing single cell RNA-seq. So then we could integrate this and learn what the T cells and B cells are secreting, and we could learn if this is actually from a clonally expanded T cell or non-clonally expanded T cell. When you look at just the overall repertoire, so in this figure, the size of the circle, these you could see like inside here that there's some big circles, and then out here this looks like a dark mantle, but this is comprised of just single dots. So as the dots coalesce, you have more of that particular clone. So this would be a large T cell expansion, and these would be individual non-expanded T cells in the cortex here. You can't really see I'm sorry. I wish you could see it, but the center of these, you see clusters of cells together. Those represent clonal expansions, and the mantle represents unexpanded T cells. You could see that PBMC, the peripheral blood, looks way different than the skin. Like in the skin, we have much fewer single T cells, unexpanded T cells, versus the blood, we're chock full of unexpanded T cells. So now we could integrate this TCR sequencing with the single cell sequencing that we just saw, and we could look at what are the most expanded T cells and what are they expressing. So when we look at necrobiosis, we could see that those individuals have high expansions of NKG7 T cells. NKG7 is a marker for like very activated CD8 T cells. Interestingly, oh, here we could see it for some reason now. IL-7 receptor is a receptor that's IL-7 is used for T cell survival. We could see that these expanded NKG7 have very low IL-7 receptor, but they are expressing other things that are probably from recently activated or chronically activated T cells, like they're expressing chemokines, and they're expressing lots of this TNF-SF9, which is for survival of activated T cells. They also have other NK receptors like KLRC3. So we could do the same thing for the B cells. And it turns out that some of the most activated B cells, some of the most expanded B cells are those IgM expressing B cells. And we could tell that they're plasma cells by their expression of different markers for example, this marker here, this gene encodes CD20, which is lost on plasma cells. And you can see that the expansion doesn't have those cells. So these are other plasma cell markers. So very weird that the skin in NL is chock full of these IgM expressing plasma cells. Now that's fine, but what about how are these cells interacting with each other? So with single cell sequencing, we could do this receptor ligand interaction. So you could see that, oh, these cells are expressing particular cytokines or chemokines or whatever effector molecule. And then you could find the cell that has the receptors for those ligands, those chemokines, cytokines, or effector molecules. So then, because these cells have the receptors and these cells are secreting the molecules that bind to those receptors, we could then infer that those cells are communicating in the skin. And doing that, we could see that and then we could rank the pathways that are being activated and the cell types that are having the outgoing and incoming signals. And what that reveals is that these SP1 macrophages are a dominant cell type. They have the most outgoing and incoming signals. And this SPP1 pathway is the dominant pathway that has the most outgoing and incoming, is providing the most outgoing and incoming signals as well. When we look at the individual cell types, we see that everybody's talking to each other. So these NKG7 T cells are talking to many of the other T cells, especially these CCL5 fibroblasts. These CCL5 fibroblasts are talking to like every single cell type. Activated B cells are talking to the CCL fibroblasts and other cell types. These SPP1 macrophages have strong communications. The other way to look at this is just to look at the specific signals. So what is TNF doing? What is SPP1 doing? What are like the chemokines doing? And you could see just by looking at by the individual pathways that there's a lot of communications going on between these cells. So from that, we can make this cartoon of what we think necrobiosis is. We have in the center these SPP1 macrophages, these osteopontin secreting macrophages. Those are talking to lots of different cell types. We have some T cells that are making lots of TNF. We have these NKG7 T cells that are making interferon gamma. All of these cells seem to be making things that cause fibroblasts to differentiate into CCL5 secreting fibroblasts, which in turn are feeding back onto all the other cell types. Ultimately, we're getting deposition of a ton of different types of collagen. So here we have collagen 6A3, collagen 8A1, collagen 11A1, and all of these collagen 4 molecules. We're also getting other extracellular matrix proteins like tenacin, postin, and fibronectin. We are getting a bunch of matrix metalloproteinases secreted. So this collagen is getting degraded, but we also have a very high upregulation of inhibitors of matrix metalloproteinases. So I don't think this is just simply a disease of collagen deposition. You're actually having altered extracellular matrix being deposited. This all ties in finally to having a bunch of C1qs and a bunch of IgG and IgM being deposited in the skin as well. So well, what about just targeting osteopontin? It's already been implicated in many rare diseases, especially things like fibrotic diseases like pulmonary fibrosis. It also, this very classic paper from 2000 published in Science, it drives type 1 immunity. So a lot of that interferon gamma is probably secondary to there being osteopontin here. There's no trials for osteopontin, but there's lots of drugs highlighting it as an important target for the treatment of fibrosis. But it's going to be very difficult to target osteopontin because osteopontin has many different domains and could actually bind to extracellular matrix proteins through an integrin binding domain. So it's not really like inhibiting IL-13 in atopic dermatitis or inhibiting IL-17 in psoriasis. This guy is fixed to the extracellular matrix, and it has lots of other functions in physiology like bone remodeling. So there's going to be off-target effects if we target it. There's also lots of different splice variants. So like which is the best splice variant to target? CCL5 has been known to be a driver of fibrosis in many different settings, like interstitial renal disease, lung fibrosis, tons of different papers on how CCL5 is driving fibrosis in these different disease settings. So that seems like a reasonable target. And luckily, there's a bunch of drugs that already exist that target CCL5. That's because its receptor, CCR5, is one of the HIV co-receptors. So there's a lot of drugs that were developed for HIV that are being tested in HIV that we could try to repurpose to see if they'll work in NL. Of course, we have these JAK proteins, these JAK and TYK2, which we could also use. However, that would only be like working for interferon. It won't be touching all of these other chemokines that seem to be important in the disease, which is probably why we're not getting like a slam dunk when we use these drugs to treat. I probably suggest a multimodal approach with numerous drugs, some topical therapy in addition. So in summary, necrobiosis is due to altered secretion of extracellular matrix proteins, especially increased expression of diverse collagens and extracellular matrix processing enzymes by fibroblasts. And fibroblasts likely play an important function by secreting all these chemokines to support like B cells and neutrophils and macrophage migration. Potential drugs, off-the-shelf ready JAK inhibitors look like they have some promise. IL-6 inhibitors haven't been really tried yet, but those IL-6 is another pathway elevated in NL, which I didn't talk about. And then we have drugs that are in development to like IL-32, CCL5, CXCL9. So those might be interesting. Osteopontin is a difficult target. So now in the last few seconds, I just want to talk about spatial sequencing because spatial sequencing is going to totally change the way we understand all these dermatologic diseases. Spatial sequencing, just like single cell sequencing, is rapidly evolving. So remember I told you we could only detect like 7% of the transcripts in single cell sequencing. Before we could only detect, we could only sequence areas of a slide for spatial sequencing. So spatial sequencing is basically sequencing similar to RNA sequencing, but performing it on a slide. This is the latest technology. So the latest technology is the Xenium, and it has single molecule resolution sequencing on the slide, whereas just like 2 years ago, if you look at my publications, we have papers on spatial sequencing, but we're using a technology that sequenced areas of a slide. And now we're down to single molecules, single transcript molecules. And in 2 months, we're going to be able to integrate this with single cell proteomics too. So it's rapidly evolving technology. This uses an amplification system, so it's highly sensitive. This is also 10 times more sensitive than other single cell sequencing technology. So, and it does this by amplifying the signal. One downside is it takes forever to do a slide. So I have this machine. So the machine's incredibly expensive, and I have it running 24 hours a day, 7 days a week, because it takes about 6 days to run 2 slides. It produces these very beautiful images. These are images of skin. You can see the epidermis, dermis. You could color them different ways. But what's really interesting is that you could now not only see the anatomy, the histology of the skin, but you could also look to see what each cell is actually producing in terms of expressing in terms of transcripts. This is Necrobiosis lipoidica. You could kind of recognize it on its own, but what's cool here is that we could actually go into the cells and figure out what each cell is making. Now, it uses, so this is one of the granulomatous areas of the slide. It uses this technique. So here, it looks just like a bunch of cells smudged together, but it uses this very advanced technique, 4 different methods to identify the cell boundaries. Because once we have the cell boundaries identified, then we could see how the cells are interacting with each other. And that's what really puts this technique apart from other techniques. So now it's the same image, but here we've outlined the cell boundaries of each cell, and visually you can see it does a pretty good job doing that. Here's another closer up image, and you could see it fairly well, that the boundaries are pretty well defined. From this, you could do normal stuff like you do with single cell sequencing. You could just look at all the different cell types and seeing what they're secreting. And this is necrobiosis. So here, I'm just highlighting that in healthy skin, you don't have the fibroblasts transcribing much collagen 4 or collagen 11, but these are highly upregulated in necrobiosis. And then we also know the position of the transcripts. And because we know where the transcripts are present within the cell, we could do some really advanced bioinformatic analysis. So this is a technique called RNA velocity, and this is traditionally done with single cell sequencing by looking at the ratio of pre-RNAs to mature RNAs. This will tell you how fast the cell is transcribing a transcript. Like if there's a lot of pre-RNAs, that means it's upregulating that transcription. The velocity is going up on how fast it's making that transcript. But we could also we've also adopted this to spatial sequencing by predicting where the nucleus is. So here we could let me go back. Well, it's not showing my pointer, but here we're using a distance formula and looking at where the transcript inside the nucleus or outside the nucleus. And by doing that, we could then map that back onto a two-dimensional image showing where the cells are most transcriptionally active. And here we're doing it on the right here, where you're seeing the darker cells being more transcriptional, having more RNA velocity than the lighter cells here. If we map this into vectors, you could see that these cells seem to be emerging from one group of cells to the other. These are keratinocytes, and we could do this RNA velocity for a set of genes, or we could do it for all genes, but the image is somewhat similar. Now, if we map this back onto the actual histology image, we could see where the cells are going in terms of where are they being derived from. And this is very evident from the epidermis here. We could see that the keratinocytes are moving from the bottom of the epidermis up, and that matches their RNA velocity vectors. Now, the other thing we could do is remember when we talked about single cell sequencing, we were talking about receptor ligand interactions. And if this cell is making the ligand and this cell has a receptor, we're just assuming that they're talking to each other. But with spatial sequencing, we actually can know that they're talking to each other because we bring in the position. So the cells are close to each other, and you see one cell is making a cytokine, for example, and the other cell having a cytokine receptor for that. But you also know what downstream events are induced by that cytokine, chemokine, whatever ligand. And we can look to see if those cellular changes are happening in the cell. So this really, really is strong evidence that not only is there a cytokine receptor pair, but actually there's some kind of functional change that's happening to the cell with that receptor. So even in this is looking at those necrobiotic lesions, and this is showing how the macrophages are interacting with the fibroblasts. And even though, so in here, the red dots are macrophages, and this is internal software that we developed. So this is a representation of the actual image. So this isn't the actual xenium image, but we're using that data and putting it on here. So spatially, it's the same. And we're instead of representing the cells with those beautiful images, which take up a lot lot of memory and makes computation a little bit more difficult, we've replaced them with circles. And we could see that the macrophages here are interacting with the fibroblasts. And this is just represented by a single arrow. But really, in reality, we know exactly what that arrow represents. We know it's like what cytokine is going there and what changes are happening to the other cell. So there's much more information than that we have than is presented in these figures. But what I want to emphasize is that a lot, not like every single macrophage is talking to the fibroblast, but a lot of the macrophages are communicating with the fibroblasts that are nearby. And you can see that one macrophage is sometimes, multiple macrophages are sometimes communicating with one fibroblast, and sometimes one macrophage is communicating with multiple fibroblasts. And the same thing goes with the T cells. It's not surprising because these T cells are making interferon gamma and other things. And so here the T cells are red and the macrophages are blue. And you can see that one T cell is interacting with multiple macrophages. Again, we know exactly what these interactions are, but we're representing them just with an arrow here. And there's even some B cells interacting with macrophages, although not a lot. So in summary, spatial sequencing will revolutionize our understanding of the pathophysiology of skin diseases. I just gave NL as an example, but we're rapidly doing this to all dermatologic diseases that we could get our hands on. Okay, thank you so much. I hope I didn't go over.
Much. Let's have some questions. Mine is sort of blown by all the data that we saw. One question was for spatial transcriptomics, that was off of a paraffin,
Yeah.
embedded slide.
Yeah, so
Yeah.
again, we couldn't do this like a few years ago, but now we could actually do single cell sequencing or single cell like sequencing from paraffin tissue.
Yep.
We could go back through all of our archived tissue. And then the other thing is we could do this very advanced spatial sequencing.
Okay.
From paraffin. And the tissue could actually be pretty old, like 5 years old. It's okay.
Okay. Yes, Dimitri.
Hi, really exciting, interesting talk. Do you have any data on patients that are responding to therapy? Yeah, looking either with spatial or single to see if the populations of cells that you think might be involved are diminished or changed?
Not for NL, it's a rare disease, but we've done that for just about like all the common dermatologic diseases. We're doing that on and off therapy and the changes that are associated with one therapy versus another therapy. So those will all be coming out like this year and next year. Yes. Yeah, so we did one active patient, and then we did multiple archive patients. So we have that. Remember, I said we have two techniques. One is single cell sequencing, which is done on fresh tissue, and then we have the same method that we could do it on archive tissue. So we did one patient fresh, lesion, non-lesion, and then we did like 12. I don't know how many. It's in the paper, but we did many patients archived using a very similar technique to single. And we did NXG, NLD, and then we also did bulk sequencing on fresh tissue from like 3 patients.
Yeah, because when we did similar approaches with suites and PGs and so forth, there were some commonalities, but we were struck by some of the variation, the results we'd see between individuals, and it looked like you were presenting data from a single patient. I just wonder if you had.
We have so in the paper there's the data from one patient, and then there's the data from I'm sorry, I think it's 8 patients, and it's all the same. Yeah, it's all the same. And I think that the reason why there's so much difference in PG is that there's such a big heterogeneity in what people consider to be PG. So I really don't believe, like, I think most of the papers published on PG have a lot of non-PG ulcers, and PG also differs on like its stage. So how active versus non-active, whereas NL is pretty, if it's not treated, it's pretty stably active. Although you could probably imagine some differences between like that erythematous rim or not, but when you look at the histology of NL, you characterize it. I mean, you could recognize it pretty well. If you look at the histology of PG, the pathologist is going to be like, could be PG, maybe not, or maybe it's like something else. So I doubt that a pathologist could just nail the diagnosis of PG, whereas they could nail the diagnosis of necrobiosis. And the same would be for like classic cases of psoriasis and atopic dermatitis. So yeah, but yeah, we've characterized like millions of, we have the largest number of patients that have with the classic diseases. So there is some variation, but there's strong overarching conclusions that could be made for all the classic diseases like atopic dermatitis, psoriasis, etc. So I mean, we have data sets with hundreds of people now in those diseases.
Great talk. A little simpler question. For the spatial seq that you're demonstrating,
That was for multiple patients, by the way. The
Great.
spatial sequencing was from 5 patients.
I was curious if specifically the populations of macrophages that were talking to the fibroblasts were a specific subset that you found in that. Like for instance, the SSP1 positive macrophages. And then
Yeah,
is
exactly.
that correct? And then the follow-up question is, do you think that the macrophage, those SSP1 macrophages are really driving the disease? Because it also looked like the CCL5 fibroblasts were communicating with a lot of different immune cells as
Yeah,
well.
no, I mean, I think that the CCL5, as I think Dr. Gallo is also on the team, that fibroblasts are really important in skin inflammatory skin diseases, that they're somehow part of the immune system. So I really think that fibroblasts are playing an important role. But when we did those outgoing, ingoing signals, we could then rank them based upon who's contributing the most outgoing signal, who's receiving the most incoming signals, and those the SSP1 macrophages were the top cell, and then the SSP1 pathway was the top pathway. So that's like that's all done hierarchically without any like peripheral understanding of what's happening, and it just popped out that way. So I do think that SSP1 macrophages are like the central to this, but I like the idea that fibroblasts are somehow driving the disease too.
Thank you.
When you said that you overlaid like sarcoidosis data with NXG, did you find any similarities? It'd be nice to know if like the whole granuloma formation had
Yeah.
some clear signals.
Yeah. So in exactly. So when we looked at systemic sclerosis, and it was quite different from NL, but sarcoidosis had a lot of similarities. Sarcoidosis also had SSP1 high macrophages, but they weren't like as prominent here. And both NL and sarcoidosis had a lot of interferon gamma, but like the interferon gamma Th1 side of sarcoidosis was just like much higher than NL. So I would say that sarcoidosis and NL are very similar, but not, this like the granulomatous disease of sarcoidosis is strongly driven by Th1, whereas it's more interesting in NL.
And osteopontin wasn't overexpressed in.
Yeah,
sarcoid?
it's also
It's also-
overexpressed, but not.
Is there a mouse model of osteopontin?
There are mouse models. I'm positive because of that paper, I believe was a mouse,
Yeah.
where they showed that osteopontin was a driver of Th1 disease, or Th1 cells, T cells. So I'm pretty sure, but I'm not positive to tell the truth. Yeah, so I think spatial sequencing, like within like maybe even a year, we'll be able to diagnose every single disease in dermatology. So yes, absolutely. But the problem is it's not going to make them obsolete anytime soon because it's just it's too expensive and it takes too long. Yes, yes, it's $1 gazillion. So you guys have some job security until the technique becomes faster and more affordable, but it's way too cost prohibitive now. But absolutely, it will be able to diagnose everything.
Thank you again.
Oh, thank you.
All right, so this concludes our rounds. Thank you all.